Multiple sequence alignments can be obtained by using those profile-based methods. A profile is where we take those alignments and we convert them into PSSMs, which are position-specific scoring matrices, and we can use these profiles in order to do some multiple sequence alignment. So here, for example, we are seeing that there are transitions between different nucleotides and there are traversions. What are transitions? These are the substitutions from same group, that is purine to purine or pyrimidine to pyrimidine. So if in this matrix we see that if we have an A on one side and A on other side, we come up with a score which is +2, and we have different scores. For example, if A replaces C, and that is -2, because A is coming from these purine groups and C is coming from pyrimidine groups. So whenever we know those nitrogenous bases, they are divided into two groups, adenines and guanines, which go as purines, and we have cytosines and thiamines, they make up the pyrimidines. So same way, since these are the variables which give recognition to our DNA, and DNA nucleotides are recognized because of these nitrogenous bases, so we can classify DNA nucleotides based upon these nitrogenous bases. So here, if we have an A nucleotide and that has been matching with an A, obviously it's an exact match, so you get a +2 score. So if we have in this example, if we have a change where we have a transvariant where we see a combination between A and T or C and G, so in that case you will penalize it more. So sometimes we take those profiles, we go into the data sets, which are kind of, we call it as training data sets, or we go to the databases where we record what is the frequency of those different amino acids on different positions of those proteins. So we can use that information, profile information, to validate our alignments. So amino acids at each positions in the alignments are scored according to the frequency with which they occur in nature or databases. We can use substitution matrices, and in this way we can add up the evolutionary distances and we can weight the overall scores of these alignments. There are mainly probabilistic methods in statistics which are used to generate those profiles. One of the most famous example is use of hidden Markov models that can be used to build up the profiles. So for example, here we are building a profile for a sequence that can be coming from, say, 4 columns or 4 nucleotide positions. So we can start anywhere. For example, here there is our begin state, and we kind of insert a sequence somewhere. We start with a sequence, and then from there we can come up with to a state where we have a match state. So M1, M is match, I is for insertion, and D is for deletion. So and then these different shapes, they are like representing your deletions are represented by those red circles, and where diamonds they represent your insertions in this diagram, and your squares they are your match states. So remember, we are in match state, we are also putting the mismatches also. So it can be match or a mismatch. So anywhere you find two sequences where at some particular column you get nucleotides on both the sequences, so that case is which is our match state. So it can accommodate mismatch also. These edges are arrows, they represent your probabilities. So transition probability is an arrow, and this kind of a half circular arrow is your multiple residue insertions, or where you can get large number of residues inserted, and then you go further where you can get some match point. So in this way, system can move from one state to another, and all the states which are possible they have been put into this kind of a model. So here is one example. We try to get a pathway for sequence like NKYLT. We get an alignment like this. So we have in column number 1, all 3 sequences have N, 4th one has got Q, column number 2 where we got an insertion and deletions on other sequences, whereas insertion of K in third sequence. And then we have a couple of mismatches in this third column or third nucleotide position and so on. So we try to model this NKYLT, which is actually this sequence number 3. We model it like this. So from beginning we go to a M1 state, there is a match state, and from there we go into an insert state or I. And then we move to M2, M3 and M4, and finally we end it. So what is the probability of starting in an M1? So from beginning you can get to any one of the 3 states, either D1 or M1 or I1. So in that case the equal probabilities, that is 1 out of 3, it's 0.33. And then you can have a insert state with where you can have a probability of seeing an amino acid K. So there is an insert state, but within that insert state, again from the beginning you can see any one of those symbols. So in the beginning you can see the symbols like it can be inserted over there. It can be any sequence coming from 20 amino acids. So the probability of seeing that sequence, which we call it as in hidden Markov model examples, we call it as emission probability. So you are in any of these states. There are 3 states in the system, M, I and D. So probability, the transition probability of moving into those states or staying within those states is 0.33, since there are 3 possibilities where our system can go. But within each state, what is the emission probability? There are 20 amino acids, so the emission probability is 1 by 20, which is 0.05. So we take all those probabilities, emissions and transition, and we multiply them with one another, and finally we can come up with the overall probability of this sequence, which is NKYLT, that is equal to 6.1 into 10 power minus 10. Profiles in case of nucleotides or in case of the DNA, we can represent them as the frequency of nucleotides in the databases or in our training data set, where we can have a set of different sequences. So for example, here on position number 1 or column number 1, we have total 8 columns in this alignment. So on column number 1 we have, most of the time we see A's, that is 72% of the time, and then 14% each we would see T's and G's, and we don't see C's in this column. Same way we can see that in the second column we are seeing T's which are 4 times more than any of the other ones, A's or G's. We see more G's in third column and so on. So in this way we can generate these profiles, and we can put those profiles in our record, and then we can come up with some alignment, and then we can align those sequences, and for those aligned nucleotides, then we can look back into our profiles whether they are following or fitting to our profiles or not. This kind of arrangement is good if you are trying to find out some motifs or patterns within your DNA or within your proteins. So those kind of databases like motif databases or pattern finding databases where you have those protein families or those motifs, this kind of profiling schemas may be important. So we compute multiple alignment given the profile HMM. We run the algorithms, Viterbi or something, and then we indicate which we can come up with the most successful path to our sequences. The issue with this profiling method is our results are skewed, and mainly they are fitting to our training data. So they are kind of, limitations, or it's the main drawback of overfitting.